And without further ado, I would really like to introduce our next speaker, Kamil Kranc from the Barts Cancer Institute in the UK. And the title of his talk, The Impact of RNA Modifications on Normal and Malignant Haematopoiesis. I just remind you, if you have questions, please put them on the chat. Kamil, when you're ready, thank you so much for joining us today.
Thank you so much. I'm just about to share the screen. Is it visible?
Yes, it is.
Okay, excellent. Thank you so much for inviting me and giving me the opportunity. It's great to be with all of you virtually today. My name is Kamil Kranc. I'm a chair of hematology at Barts Cancer Institute in London. And the central aim of my lab is to understand molecular mechanisms controlling normal and malignant hematopoiesis. And today I will talk to you about the impact of RNA modifications on those processes. Hematopoiesis serves as a paradigm for our understanding of cellular hierarchies, normal tissues, and cancers. Hematopoietic stem cells possess unique cell renewal capacity and multilineage differentiation potential and give rise to all blood lineages. And due to those specific properties, those unique properties, stem cell transplantation offers the best chance of curing many diseases, such as blood malignancies, bone marrow failures, autoimmune diseases, and genetic disorders. However, there's a shortage of stem cells for transplantation purposes, and we must devise the ways of expanding stem cells for widespread transplantation. Acute myeloid leukemia, on the other hand, is a clonal aggressive disease of stem and progenitor cells, which acquire diverse mutations to generate leukemic stem cells, which fuel leukemia. Currently available chemotherapies normally damage normal hematopoiesis, but they fail to eradicate leukemic stem cells, which prevail and cause relapses with severe consequences to the patient. So the central aim of my lab and many other labs is to address this unmet clinical need in AML by pioneering brand new therapies against novel targets to selectively eliminate leukemic stem cells in AML. It is well known by now that the strict regulation of gene transcription is essential for hematopoietic stem cell integrity, and the dysregulation of transcription results either in stem cell failure or leukemogenesis. But what has been overlooked for a long time is that RNAs, in this case mRNA transcripts, undergo chemical RNA modifications. And the significance of many RNA modifications in normal and malignant hematopoiesis is really poorly understood. And this obviously gives us opportunity for brand new discoveries with a potential therapeutic impact. So this is RNA as we know it, mRNA. It has a coding region flanked by 3' UTR and 5' UTR, starts with a cap, finishes with poly-A tail. But what has been known for quite a while, but really ignored functionally, is that almost every single residue within RNA could be chemically modified. In this case here, for multiple reasons, I will focus my talk on RNA modification, which is referred to as M6A modification. We ask the question, what is the functional significance of this modification in AML and normal hematopoiesis? M6A is the most abundant internal RNA modification in which adenosines at position 6 are methylated by enzymes called writers, and this methylation could be removed by erasers. And the function of M6A is executed by proteins called the readers, which specifically recognize M6A modified transcripts to promote RNA decay, translation, or splicing. We have focused on 3 cytosolic readers, YTHDF2, which promotes RNA decay, and YTHDF1 and 3, which promote translation. However, more recent data suggests that they could be redundant and they may all promote RNA decay. We found that YTHDF1, 2, and 3 are highly overexpressed across different AML subsets. And we also found that YTHDF2 and 3 expression is correlated with leukemic stem cell activity. Because of the high expression of YTHDF2 transcript and also protein in diverse AML subsets, we focused on YTHDF2, and together in collaboration with Donald O'Carroll in Edinburgh, we asked the question, what is the impact of YTHDF inactivation on leukemogenesis? So to address this, we took advantage of a mouse model system where stem and progenitor cells from mice are transduced with multiple different naturally occurring oncogenes. They are serially replated to generate pre-leukemic cells, which upon transplantation to mice generate leukemia. So we performed this model with wild-type mice and mice lacking YTHDF2, where the gene is missing, specifically in the hematopoietic system. And what we found was striking. No matter which oncogene we have transformed the wild-type and knockout cells, we found that the YTHDF deficient cells fail to undergo efficient transformation in culture. And when we took those pre-leukemic cells, which are wild-type and knockout for YTHDF2, and we transplanted them to a recipient mice, what we found is that YTHDF2 deficient cells very weakly established leukemia and developed disease only with a very delayed latency. So YTHDF2 deletion is very significantly extends latency of AML. We have also shown that YTHDF2 is required for leukemia propagation. I don't have time to discuss this, but we can talk about it later. So does this have any significance to human? So we took, to start with, multiple human cell lines and AML cell lines derived from patients and knocked down YTHDF2 expression. We can see that the knockdown resulted in decreased proliferation and increased apoptosis of human cell lines. So then we went to primary human patient samples, and we knocked down here. We are showing 3 different examples of 3 different patient samples. We can see that the knockdown of YTHDF2 almost completely obliterated the ability of these patient samples to form colonies in culture. So then we wanted to understand the molecular mechanism through which YTHDF2 inactivation compromises AML. We performed RNA sequencing. As expected, many transcripts should be upregulated if you delete the gene which is required for RNA degradation. We also performed M6A sequencing, and combined analysis of this indicated that there are many significantly upregulated genes, and those upregulated genes upon YTHDF2 deletion have enrichment of M6A occupancy. So this is what's expected. We found that inactivation or deletion, in this case of YTHDF2, upregulates many transcripts that are M6A modified. So then we wanted to interrogate these transcripts against multiple diverse signatures in different diseases. And without going into technical details, what we found is that YTHDF2 knockout cells have increased expression of those genes whose expression is also upregulated in patient samples, but those patient samples which have lost the ability to propagate leukemia upon transplantation. So in other words, if you inhibit YTHDF2, in this case, delete, we get upregulation of M6A modified transcripts that are associated with a weak leukemic stem cell activity. And one of those transcripts that we found was TNF receptor 2. TNF receptor 2 transcripts are very highly methylated, both in mouse and human. And this is just a cartoon to show that YTHDF2 recognizes M6A modified TNF transcripts, TNF receptor 2 transcripts, and this results in degradation. TNF receptor 2, as you know, is a cell surface molecule, so we can detect it by flow cytometry. And indeed, if you delete YTHDF2 in leukemic cells, we can see a massive upregulation of TNF receptor 2 on leukemic cells. So then we ask the question, are these cells sensitive to TNF receptor, TNF, tumor necrosis factor? So we incubated wild-type and knockout cells with TNF, and what we could see is that indeed the TNF, the YTHDF2 mutant cells really apoptosed much faster than the wild-type cells. So we think that increased expression of TNF receptor 2 upon YTHDF2 loss might sensitize AML cells to TNF. So we think that YTHDF2 could be a potential therapeutic target in AML, where when you inhibit YTHDF2, this compromises leukemic stem cell activity and rescues mice from disease. But if this was supposed to be a good therapeutic target, well, one would hope that it doesn't have much impact on normal hematopoiesis. So we asked the question, what is the impact of YTHDF2 inactivation on stem cell biology and hematopoiesis? So this is how the blood looks like in young mice lacking YTHDF2. We can see that they have a slight decrease in white blood cell counts, possibly very mild anemia, but these mice age very happily, live for more than a year and a half, and don't really show any adverse effects. But what was very consistent that we found in young and also old mice is expansion of hematopoietic stem cells and diverse multipotent progenitor cells. So one of the ways to test stem cell activity is to transplant them into mice. So we took wild type and knockout hematopoietic stem cells and transplanted them to primary recipients. Overall, they reconstituted hematopoiesis just fine. We had enhanced myeloid reconstitution, normal B cell reconstitution, and decreased T cell reconstitution. But if you look into the bone marrow of those primary cells, we can see that stem cells lacking YTHDF2 very robustly reconstituted stem cells and progenitor cells, undifferentiated cells within the bone marrow of the recipients. So then we took stem cells from primary recipients and retransplanted them to secondary recipients. And what we observed was very striking. We could see that stem cells lacking YTHDF2 almost completely lost the ability to generate muciliage hematopoiesis. So we think that in the longer run, YTHDF2 protects stem cells from exhaustion. So then we did RNA sequencing to understand some mechanisms, and we only found 81 transcripts being upregulated in stem cells who were lacking YTHDF2. But what was really striking is that the majority of those belong to a pro-inflammatory transcript, pro-inflammatory pathways such as interferon gamma, alpha, interleukin signaling through STAT3, TNF signaling. And we are very excited about it because, as we know, pro-inflammatory signals result in stem cell exhaustion and inhibit cell renewal. So then we performed M6A sequencing and integrated all these data together. And what we find is that multiple transcripts that are upregulated and belong to pro-inflammatory pathways are indeed M6A modified. And here is an example of the key pro-inflammatory transcripts. You can see that they are massively M6A modified. So we think that YTHDF2 suppresses pro-inflammatory signals to promote cell renewal of stem cells and inhibit exhaustion. But this YTHDF2 on its own, itself induced by pro-inflammatory stimuli. So we took advantage of a reporter mouse where we have our GFP fused to YTHDF2. We injected these mice with poly-I poly-C, which induces inflammation, sterile inflammation. And indeed, we can see that YTHDF2 in stem cells and progenitors is very highly upregulated upon pro-inflammatory stimuli. So what we think is going on is that in the presence of pro-inflammatory stimuli such as infection or inflammation, pro-inflammatory transcripts are upregulated, and this results in emergency hematopoiesis, where stem cells are activated and respond to the infection. And YTHDF2 is one of those transcripts, one of those proteins that is upregulated and functions as a negative feedback loop to dampen the exuberant pro-inflammatory pathways. But what happens is that if you delete YTHDF2, those pro-inflammatory transcripts are no longer degraded. And instead of driving emergency hematopoiesis, indeed, they enhance stem cell aging and loss and cause the loss of stem cell activity. So we think that YTHDF2 is indeed a therapeutic target in AML, where there is an ample therapeutic window for inhibition of YTHDF2, at least in the shorter term. But in the longer term, if we were to inhibit YTHDF2 for a very long time, then that would possibly result in stem cell exhaustion. So how can we target YTHDF2? One way to target YTHDF2 is to disrupt its binding to M6A modified transcripts. So then we ask the question in unpublished data, is in a published study, is targeting of the YTHDF domain of YTHDF2 sufficient to inactivate this protein? So here we can see a structure of YTHDF domain where you can see 3 tryptophans forming aromatic pocket that embraces and strongly binds the M6A modified oligo RNA. And what we did in collaboration with Donald Carroll is to generate a mouse model where we when we substituted, we mutagenized this tryptophan, which is required for strong binding of M6A. We mutagenized it to alanine. And we know that this biochemically disrupts the binding between M6A modified transcripts and YTHDF2. And then we asked the question, what is the impact of genetic disruption of the YTHDF M6A binding on normal and malignant hematopoiesis? So we generated 3 types of mice: wild-type mice, the mice that completely lack YTHDF2 in the hematopoietic system, and those mice that we refer to as WA, which have this mutation that disables the binding to M6A. And to cut the long story short, we can see that the stem and progenitor cells are expanded in knockout mice, but we can also see the expansion, very similar expansion in WA mice. As you remember from the previous slides, I showed that a knockout of YTHDF2 results in expansion of diverse multipotent progenitors, MPP1, 2, and 3, 3 and 4 populations. And we can see in general that those populations are also expanded if you just disrupt the binding. So we think that the YTHDF2 function is dependent on the M6A binding in normal hematopoiesis. How about leukemogenesis? So we collaborated with Jörg Schwalle, who gave us the MLL inducible mouse model, which we crossed to the WA mice. And I'm showing you the outcome of the in vitro assays, where basically we generated MLL inducible mice that are wild type or have WA mutation. And in the presence of doxycycline, which induces MLL-AF9 knock-in, we did the proliferation assays and CFC assays. We can see that upon serial replating, WA mutation almost completely obliterated the ability of these cells to form colonies. And if you perform proliferation assays, you can see that these cells basically stop existing. They don't proliferate. And at the end of the assay, they actually undergo apoptosis. So we think that the binding of M6A to YTHDF2 is critical for the function of this protein. So I'd like to stop here by saying that we think that YTHDF2 is a good therapeutic target. The disruption of the YTHDF2 M6A binding compromises leukemic stem cells in AML and results in stem cell expansion. This is exactly what you would like to achieve in AML patients. So obviously we are very excited about targeting YTHDF2 and drug discovery programs to target this particular pathway in AML and other cancers. So I will stop here. I'd like to thank my hardworking group in London. I have mentioned Don Roccaro, and Chris Schofield is our chemistry collaborator in Oxford, who helps us to develop drug discovery against YTHDFs, and obviously Cancer Research UK and various other charities, including Barts Charity in London, for funding. Thank you so much for your attention. I'm happy to take questions.
Thank you so much, Kamil, for such a wonderful talk, and also for sharing some of your unpublished results. This is very, very nice. I'm trying to see whether there are some questions on the Q&A. But meanwhile, the questions are coming. I would really like to ask your thoughts. So you've shown us the impact of this mRNA modification on tumorigenesis in hematopoietic stem cells. I was wondering how much do you think this is applicable to other tissues? I'm thinking on tissues where you have also to activate that, like where there is also inflammation related to cancer, like let's say pancreas cancer. Is it anything known on this type of modifications in cancers that also related to inflammation?
Yeah, I think that the literature in this field is really growing and proliferating every day. I can clearly see that YTHDF family, including YTHDF2, is required for breast cancer, for glioblastoma, pancreatic cancer, for esophageal or gastric. So I think that the literature is growing every day, and if somebody manages to come up with a molecule that inhibits YTHDF, that will not only be applicable to leukemia or multiple myeloma as published, but also multiple other cancers. So yeah, I think there's a huge scope, therapeutic scope here.
I fully agree with you. There is one question coming from the audience, from Julie Panikin. How do you explain the effects on mRNA modifications could have opposite effects according to the organ? I am thinking of a study that has been performed in colorectal cancer on M6A.
I'm not aware of colorectal cancer in this context, I must say. I think that
she wrote CRC, maybe she didn't mean colorectal cancer. It was my interpretation, but I guess that's what she meant.
Yeah, I think it depends on the target you are looking at. I'm aware of various papers from Chuan He and other experts in M6A field, where, for example, the writer of M6A has been shown to be oncogene, in some cases tumor suppressor. I don't know. I don't think it's very well defined what the mechanisms are, apart from the statement that it's tissue specific. But in terms of the YTHDF proteins, what we find is that in any cell types, any cancer cell lines, we knock it down, we only see apoptosis. We haven't seen this dual effect where YTHDF family can be oncogenes or tumor suppressors. Haven't seen it so far.
Oh, that's interesting.
I think this function is quite conserved.
There is a somehow kind of related question from Ana Rita Carlos. She's asking, is there survival data for different types of cancers regarding mutations in YTHDF2, mutations in expression levels?
Survival. Yeah, I guess you are referring to the Kaplan-Meier curve and the correlation with YTHDF2 expression. We have looked into this. There's some weak correlation, I must say, but there's nothing. There's no smoking gun here. There's nothing. We cannot state that increased YTHDF2 expression somehow, makes the patient's treatment outcome or survival outcome worse. We haven't seen these data yet. If you inhibit those targets in those cancers, you see rather profound effect.
There is Ilaria, I see that you have your hand raised. Do you want to switch on your camera?
Yeah, thank you. Thank you, Kamil. That was beautiful. I have a bit of a curiosity question. I was very intrigued by this data that you show that you have a kind of a time frame where when you induce the knockout and it's tolerated in the normal hematopoietic compartment, but then if you do it in the long run, then it becomes deleterious. And you've shown that this is having to do with the modulation of inflammatory pathways. So I wonder whether can you use this model to understand better what really drives the hematopoietic stem cell intrinsic aging by looking at which of those pathways are basically playing a major role in that?
Absolutely. That's a very good question. We are looking into this. We are trying to dissect which pro-inflammatory pathways, which arm of pro-inflammatory signaling at the molecular level drives the stem cell exhaustion. But it's obviously it's an aging project, so it's going to take a little bit of time. I think it has to do with interference signaling. This is what we find, that interference signaling is driving this. But I get all sorts of questions regarding this, because we do know that interference signaling predisposes to transformation or to myelodysplastic syndromes. But somehow we don't see any malignant transformation upon YTHDF2 mutation in the longer run. We never saw it. So there's some discrepancy here between what we observe and what pro-inflammatory signaling is meant to do. But it might well be that it has to do with clonal hematopoiesis. Perhaps YTHDF2 deletion in the longer run will contribute to clonal hematopoiesis. We are looking into this in detail as well, but these are early days. I cannot really show you any data yet.
Thank you.
Wonderful. So that was a great discussion. I think, Kamil, if there is no further questions coming to the floor, I would like to just thank you so much for your outstanding talk and for joining us here today.